Hi, welcome to today's webcast on building green manufacturing on a foundation of PAT and QbD. I'm Paul Thomas, Senior Editor of Pharmaceutical Manufacturing Magazine, as well as pharmamanufacturing.com and pharmaqbd.com. And obviously I encourage you to visit our websites, in particular pharmaqbd.com, since it has a mission of really chronicling the movement of PAT and quality by design within the drug industry, and also really to help advance science and risk-based drug manufacturing. We'll have three speakers on today's program, and I'll be the first, talking about the connection between PAT and QbD and green manufacturing. I'll be followed up by Dominique Ebro, and Dominique is with Mettler Toledo AutoChem, talking about PAT for the greening of batch processes. And then finally, Kurt Hiltbrunner of Mettler Toledo Process Analytics will talk about intelligent sensor management and how it can aid QbD in sustainable manufacturing. And finally, we'll have a Q&A with all three of us. So please send in your questions at any time during the program, and we'll try and get to as many as we can. There have always been differing definitions and interpretations of PAT and QbD, but really QbD is much broader. Here's a definition from an FDA slide set of a couple of years ago. QbD is scientific, risk-based, holistic, and a proactive approach to pharmaceutical development. It involves a deliberate design effort from product conception through commercialization and a full understanding of how product attributes and process relate to product performance. PAT, of course, generally refers to the actual technologies that provide this product and process understanding. I like an analogy that Mettler Toledo's own Terry Redmond shared with our PharmaQbD readers recently. Terry wrote, if QbD is the map for drug development and manufacturing, then PAT is the GPS, monitoring the process and telling us where we stand at any given moment. And I'll quote from Terry. Quality by design, properly applied in the development process, should give us the process map needed to get from point A, raw materials, to point B, finished product. And then he continues, but the only way to be absolutely sure we are on track is to be able to directly measure our true position in real time, our critical quality attributes. And this is where analytical technologies become so important. This distinction between PAT and QbD is really important in discussing how they might leverage green and sustainable manufacturing, and I'll touch upon this more later in the program. FDA and other regulators, of course, would be more than happy to have PAT and QbD serve as a foundation for sustainable manufacturing and greener practices. FDA lately has been working much more closely with EPA and other organizations to ensure the environmental sustainability of drug manufacturing. And issues of sustainability and things like this are not necessarily found within FDA guidance or within ICH guidance and those guidances that are foundations of PAT and QbD. But certainly there is language in them that dovetails with the ideals of green chemistry and engineering, industrial waste and energy reduction, the reduction of corporate carbon footprints, and so on. Here's a snippet from FDA's PAT guidance, and I think efficiencies is the key word here. In general, you might say that efficient manufacturing translates into greener and more sustainable manufacturing. That is, fewer materials are used, equipment runs for shorter periods, factory shifts are shorter, lights can be turned off in offices and factories earlier, and so on. So more importantly, really, months and even years of wasteful development activities can theoretically be trimmed by using PAT technologies and adhering to QbD practices. The well-known principles of green chemistry and green engineering also dovetail nicely with PAT and QbD. For example, these two green chemistry principles echo some of the language that you'll find in PAT and QbD guidance in their emphasis of intelligent process design to achieve specific ends, and in the value of real-time analytical methodologies. These are just two of the principles of the 12 green chemistry principles, of course, but you can see they're reminiscent of the language found in FDA and other documents. The industry certainly, I think, Pfizer, for example, from what I've heard and read recently, is beginning to consider what can be called green chemistry metrics more in its practices. And so obviously this is something that suggests an overlap between green chemistry and PAT and QbD. The same goes for the principles of green engineering, as I mentioned. Principle 4 here speaks to efficiency. Products, processes, and systems should be designed to maximize mass, energy, space, and time efficiency. Again, the more efficient a process is, the more likely it is to be sustainable from a business perspective, really, but also from an environmental perspective as well. So the question of whether or not PAT and QbD can be used to leverage sustainability efforts at drug manufacturers can seem like a bit of a no-brainer. If you've got a well-designed process and you've got tight control of that process, it only stands to reason that there will be little waste and thus production can be optimized. Last spring, we at Pharmaceutical Manufacturing surveyed our readers about their green manufacturing habits, and I like this quotation that one reader sent back. "Process control is environmental control," he said, and it seems to have a nice ring to it. Some obvious applications that I came across in terms of viewing that survey and writing an article based on that survey of ways that PAT in particular can be used to leverage sustainability efforts and to be more environmentally sound in terms of manufacturing. One of the people I spoke to, Jack Carroll, who is an industry consultant, has seen it firsthand in terms of a situation where PAT can be used to wind down a fermentation process 2 days early, resulting in what he called serious energy and cost savings. So obviously that manufacturer, by winding down a fermentation reactor 2 days early, recognized some real savings, and this is the kind of thing that PAT can do along the lines of sustainability. So we know that PAT can have some serious environmental advantages. Still, however, sustainable processes up to this point have not been high on the industry's priority list. So it's fair to ask the question, is industry on board? The question of whether or not PAT can be used to leverage sustainability was posted by someone at Genentech last spring on a LinkedIn group, the Process Analytical Technologies group on LinkedIn. A very good group if you're a LinkedIn user and want to get in on some good discussions. Nevertheless, several members from the manufacturing community chimed in about the question, and here's what one person said: energy savings and sustainability have always been secondary or tertiary considerations. Other things tend to dominate, such as process yield, purity, cycle time, things like that. And then another person chimed in and said PAT teams just have other priorities. So I think that's a fair statement, and hopefully that'll change over the years, but this, I think, is the situation right now. Could this be changing? Are we at a tipping point? I believe so. I think many major manufacturers are beginning to look at PAT to leverage their green and sustainable missions. One of the people that I've talked to in the last couple of years is Daryl Ertl, who is with GSK, and he's on their PAT team down in the Research Triangle in North Carolina. And he talked about the fact that GSK's pilot plants, in looking to reduce cycle times, save energy, and cut costs, have gravitated towards PAT. So they're really seeing PAT as something not that is just there to control and to optimize processes, but something that can be used to cut energy, for example, significantly, and have sustainability objectives. Earlier this year, Ertl told me GSK established a worldwide team to promote PAT, mainly using NIR and mass spectrometry to determine endpoints of drying operations, with energy savings as a specific objective. So Ertl has noticed a significant change within his organization, and obviously GSK is one of the bellwether manufacturers within the industry. Here's some more data from our survey last spring, which I had mentioned before. Does your facility look at PAT with an eye towards sustainability and green practices? And we asked this to our readers, and a healthy segment of our readers wrote back. I believe it was about 100 people in total, but 10% of those said yes, sustainability has become a key driver of our PAT efforts. 38% said yes, but it's not a top priority. 30% said no, there are other factors that are driving PAT. And then 22% said no, we're not incorporating PAT into our processes at all. And we would hope that that number would go down in the upcoming years. In that same survey, we asked readers which areas have the most potential in terms of PAT and sustainability. 70% said PAT has the ability to reduce failed batches and thus wasted time and materials. And that was the number one reason. 60% optimizing chemical reactions and reducing solvent and material use. 60% said reducing cycle times and changeovers. 58% reducing the time that equipment such as dryers, cleaning place equipment, and so on operates. And then 52% eliminating process steps and saving time and materials. There were some other factors in there as well, but these are the top ones. Can it happen at your plant? In other words, can PAT and QbD be used to leverage sustainability? Again, I go back to some of the things that GSK's Daryl Ertl told me. You need to have a champion high up in the company, he said, as well as the technical experts who know what they're doing. And also he reiterated that it doesn't have to be a big bang approach, but starting small and moving incrementally can pay off as well. So many small PAT projects that save energy can add up to significant savings. What about QbD? Again, another question that we asked in our survey of last spring. Does your facility company look at quality by design with an eye towards sustainability and green practices? 15% said yes, sustainability has become a key driver of our QbD efforts. 35% yes, but it's not a top priority. And all the way on down to 19%. No, we're not really doing anything QbD related. While PAT is about monitoring and control and supports QbD efforts, QbD, as we've said, is also about speed to market, which doesn't necessarily always translate into what might be considered environmentally sound practices. QbD may support that a process be designed, for example, with solvents that provide high yields, but those same solvents might produce significant waste. That is, from a QbD standpoint, the best solvent might not be the greenest one. QbD and sustainability are two different variables, not necessarily dependent upon one another, said DSM's David Ager last year, and I had a chance to talk with him in an interview, and he just felt that they can work together, but they're not necessarily always going to work together. Still, others see an organic connection between both PAT and QbD and sustainability. Manufacturers can use metrics such as process mass intensity, or PMI, to monitor and reduce the impact of their processes. And in the long run, well-designed QbD processes go hand in hand with green principles and practices. And a good resource, if you'd like to do some reading on this, is PAT and Green Chemistry, the intersection of benign by design and quality by design. And that's written by Julie Manley, Buzz Q, and John Barrage. And there's a link to that article available on pharmaqbd.com, or you can also go on ISPE's website. Let me introduce Dr. Dominique Bro, an industry expert with Mettler Toledo AutoChem, who will share two application examples that illustrate how PAT directly contributes to the design and operation of greener processes, and can greatly increase operator safety, dramatically reduce waste and inefficiency in the drug industry.
Alright, thank you, Paul, for this introduction. I really liked to see coming up at the top of the survey list the idea that pH is key and critical in reducing failed batches, therefore wasted time and material, because this ties really well into the couple of case studies I selected today. I like those examples because they are applications of PAT leading to green processes that have been proven on scale. Too often we see interesting examples of enhanced reaction conditions, but they sometimes fail to prove the point that larger scale implementation is doable. Let's start with this example from Pfizer. It concerns the development of an alternative procedure to prepare a CCR1 antagonist. The initial procedure was based on a sulfide displacement depicted here at the top. The enhanced route is based on a thioacetate oxidation using in situ generated performic acid, as you can see on the bottom figure. Although the new route resolves the great difficulty of the initial one with respect to removing sodium salt, it's an oxidation process and has the potential to release large amount of energy and gas. Now, when having to deal with a reaction presenting a potential hazard, you want to answer the questions on the left. They have to do with global and instantaneous reaction energy, as well as the relationship between reaction rate and feed rate. As you can see on the right side, they ran an RC1 calorimeter reaction and plotted specific heat flow in watts per kilo as a function of time. This plot actually contains a lot of information. First, the area under the curve represents reaction heat in kilojoule per kilo. When converted in kilojoule per mole, this represents a staggering 975 kilojoule per mole. When divided by heat capacity and corrected for dilution, delta T adiabatic is obtained. It represents how high the temperature may rise to if the reagent is added in the absence of any vessel cooling. A very high theoretical value of 170 degrees Celsius is obtained, which calls for trouble, unless the process is kept under good control. The maximum points on the curve represent the maximum heat output. It's a fairly reasonable 44 watts per kilo, probably slightly above what it should be for a power plant scale implementation. But I guess this goes against what I just said about the large value for reaction heat. The catch here is that although the reaction is very exothermic, it is perfectly dosing controlled. The reaction is fast and its rate is limited by the rate of hydrogen peroxide addition. This is a safe and comfortable way of running and controlling the process. As a result, unreacted material at the end of dosing is a mere 10%. The moral of the story is that a highly exothermic reaction is not necessarily unsafe to scale up, provided that you have sufficient evidence from the RC1 run that there is no delayed reaction onset, that the reaction is fast and can easily be run under dose control conditions, so that dosing time can be adjusted to the plant vessel cooling capacity. The process was eventually run in the power plant to prepare 30 to 35 kilogram batches. It is interesting to note that in situ real-time FTIR spectroscopy with React-IR was actually used to monitor the plant runs. This anecdote allows me to add that RC1 calorimetry is one of the methods that helps make batch processing safer, therefore greener, but React-IR is also a critical technology as illustrated in these papers. Now let's move to the second example. You know, at some point during the introduction, Paul cited GSKA and the use of PAT for the isolation phase of an API. I believe it was drying. Well, it's fair to say that until recently, scientists would spend so much time and effort improving the chemistry of the synthetic steps to end up wasting a significant part of it during the isolation phase because of a poorly designed crystallization process. There is a waste aspect to it, but also a safety aspect, as inadequate particle size results in milling, which generates fine particles, which, as you know, results in exposure and explosion hazard. So, the goal of modern process engineering is to take a broad view of process improvement and take crystallization as an integral part of the green equation. To illustrate the importance of a well-designed crystallization process, let me go over this example from Sepulchre in Massachusetts. The goal was to improve crystallization conditions of a process that would regularly fail optical purity specs at a CMO. It was also noticed that the failed batches would take longer to filter and dry. You can see on this slide a microscopic image of crystals corresponding to a bad batch. The strategy put in place focused on the study of secondary nucleation using nucleation temperature, mixing, and seed surface area as key variables. The study was conducted using a liter-scale lab reactor representing a scale down of the conditions at a contract manufacturing organization, and an FBRM probe that tracks particle size changes in situ and in real time. The batch would be seeded at 46 degrees C, but the FBRM probe indicated that seeding was not immediately followed by growth, as you can see on the figure on the upper right. It displays the rate of particle formation as a function of time and temperature. Seeding occurs at the intersection of the dotted line and the temperature profile. The rate of formation of particles remains small and erratic for about an hour, or until the temperature gets down to about 35 degrees. It was also found that when nucleation occurs early at relatively high temperature, in other words, low supersaturation, the product has a better quality and gets filtered and isolated more easily. So the key question is, how can we force nucleation to occur earlier? Alright, the figure on the right side shows a positive linear correlation between nucleation temperature and shear rate at the impeller. In other words, nucleation was limited by mixing to a certain degree. Improving mixing conditions allows nucleation to occur at a higher temperature, leading to a better quality product. You can actually see the difference here between those two pictures. Improved crystallization conditions lead to a larger, more defined crystal. Now how about seeding? In this case, surface area, not the amount of seed, seems to be critical. In other words, you don't need to have more of them, you just need to reduce the size, and that helps trigger nucleation early on, which again leads to a better quality product. Alright, here you can see the FBRM and temperature plots for two different experiments at lab scale. They differ by the mixing speed and the surface area of the seeds. The results, as you can see, is a crystallization that occurs earlier at a higher temperature, indicated by the red arrows, and generate a smaller number of larger particles. As a result, the optical purity is better and filtration takes only 30 seconds versus 150 seconds. The crystallization process was eventually successfully scaled up to a 50-liter vessel first, and then to a 400-liter vessel at a CMO. Compared to the original process, the filtration time was divided by 3, the operation was made safer, as there was no need to scrape the product out of the centrifuge anymore. The optical purity was overall better, and above all, the good results were reproducible. Overall, in situ monitoring of particle size changes was a critical tool that helped make the crystallization procedure safer and more efficient. In other words, greener. Here is a list of papers along the line of enhancing crystallization processes through the use of PAT. Some of them present state-of-the-art methodologies like supersaturation control, but making a crystallization greener does not necessarily require that much engineering, and a simple real-time visual monitoring technique can go a long way. Before concluding, I'd like to thank the major contributors to the work I just presented. Researchers from Pfizer in Groton, Connecticut, and from Sepulchre in Marlborough, Massachusetts. In conclusion, I'd like to reiterate a point Paul made in the introduction, that although they began on different paths, the principles of green chemistry and engineering share plenty of common ground with the quality by design and PAT initiatives. I hope the examples just presented helped you understand and agree that pH is a critical tool to maximize process efficiency, prevent energy waste, time waste, and material waste. Actually, I do like the commonalities between PAT and a GPS. PAT, like a GPS in everyday life, tells you exactly where you are throughout the course of the batch. PAT, used along with the process map, will guide you more effectively to the final goal of high quality standards, a little bit like the GPS guides you literally to your destination. At this point, I'm going to let Kurt from Method Toledo Process Analytics give a different perspective on the use of PAT for green chemistry and green processing. Thank you for your time.
Thanks, Dominique, for those practical examples of PAT contributing to safer and greener processes. The final portion of our webinar will be Kurt Hiltbrunner. And Kurt is a pharmaceutical industry expert from Mettler Toledo Process Analytics. He has a broad experience in the implementation of instrumentation for the monitoring and control of critical process parameters. He'll share some insight into the importance of sensor reliability and intelligent sensor management as a key element of implementing quality by design in a manufacturing environment. Detecting problems with sensors early and avoiding catastrophic sensor failure can be a vital part of risk management and sustainable manufacturing by helping to avoid significant sources of batch failure, waste, and rework. And Kurt will talk about this.
I have shown this slide to many pharmaceutical companies around the globe. So far, no company voted for the field, low control, low understanding. This would be very risky. Some companies mentioned that their process control is on a high level, but some process understanding is missing. With this situation, they have to control key and critical process parameters very tightly, e.g. the operating space is very narrow. QbD and design space approaches will allow a broader design space and more flexibility during manufacturing. The majority voted for the field, low process control, high understanding. This is a classical approach in the pharmaceutical industry. A lot of grab samples are analyzed in the lab, and it can happen that the batch is already finished when a deviation is detected. The FDA enforces the industry to adapt more inline control according to the PAT guidelines exactly due to this situation. High process control, high process understanding is the desired state of robust and reproducible processes. This webinar will demonstrate possible ways to reach these goals. The pharmaceutical industry has been very successful in recent decades, but growing price pressure, shrinking revenues, and increased R&D costs call for changes. Regulatory authorities are guiding industry towards the changes that are necessary to adopt this new environment. This slide shows a very simplified workflow from fermentation over harvesting to purification. First case study will deal with microbial fermentation, and the second is an example for a chromatography step as a purification operation. This is a typically setup for microbial fermentation in batch mode. pH, dissolved oxygen, optical density, pressure and temperature are measured with inserted probes inline and in real time. Glucose and other parameters are measured off or at line with biochemistry analyzer. This is now the three-dimensional design and operating space for the parameters pH, dissolved oxygen, and temperature. According to the principle of the ICH Chapter 8 guideline, a unit operation design space was established using the acceptable ranges for temperature, pH, and dissolved oxygen. It also can be seen that the operating space, as defined by the operating ranges, is well nested inside the unit operation design space, indicating robustness of the process step. Chromatography is a second case study today. pH and conductivity are key process parameters at the in and outlet of a chromatography column. Conductivity and pH are monitored to check the performance of the gradient, the loading of the column, the regeneration and re-equilibration. At the conclusion of the process characterization studies, data are analyzed to calculate the acceptable ranges for key and critical process parameters. These acceptable ranges together define the design space. It can also be seen that the operating space, as defined by the operating ranges, is well nested inside the design space, indicating a robustness of the process step. Electrochemical pad tools are the second largest segment according to this market survey. If parameters are identified as critical or key parameters, measuring system for pH, dissolved oxygen, conductivity and TOC are powerful pad tools. The new optical DO sensor is not an electrochemical device. ISM stands for Intelligent Sensor Management. In the sensor, a small microprocessor has been integrated that stores and processes all relevant data. All this information is then digitally transferred to the instrument without any risk of interference. The microprocessor is also able to store information like calibration values, identity, and timestamps the maximum process temperature. What is even more interesting with ISM is the connection possibilities and how the sensor can perform their own diagnostics in real time. How ISM can support bioprocesses. Keep it simple. Just plug the sensor to the transmitter. Sensor will be recognized and calibration data will be transferred to the transmitter. Pre-calibration either with iSense or with a transmitter will change your SOP. No more liquid handling in a clean zone. Pre-calibrated sensor on stock for urgent needs. Simple cable and connection with no humidity issue. Process safety. All the information from advanced diagnostics allows you to make a good decision if a sensor could use for the next batch, avoiding any sensor failure during fermentation or other processes. The iSense database contains the information about all sensor and reports can be generated if needed. The automatic counting of sterilization and cleaning cycles for better traceability. Cost savings. Commissioning with ISM system is straightforward and fast. Maintenance costs can be reduced drastically. Predicted maintenance is one of the key benefits with ISM sensors. The sensors are able to give a request when maintenance is really needed, so the maintenance can be well planned and organized in advance. The dynamic lifetime indicator, adaptive calibration timer, the time to maintenance are functions which give unique benefit to our customer. With the help of this function, they can make better maintenance planning, better process control, and even improved measuring accuracy and reduced maintenance costs. The intelligent diagnostics can help to prevent unnecessary process interruption, so ISM does not only help to save maintenance costs, it also helps to increase the operational availability of a plant. In a world of very intense competition for manufacturers, predictive diagnostic can help customers to retain and expand their leading edge and remain profitable. True Asset Management. Let me explain. On the PC or laptop that runs iSense, a complete database of all sensor operation is present, where each calibration of each sensor is available as a PDF protocol. This database can be exported and further analyzed into Excel to perform reliability trends on each measurement loop. For many customers producing biopharmaceutical, this database is the most important tool inside ISM's suite. If pH, dissolved oxygen, conductivity or TOC are identified as key or critical process parameter, ISM systems are powerful PAT tools and will allow improved process control. ISM system can be used during development and in DOE studies to define process design space. The scaling up during process development will be faster with ISM system. With ISM, maintenance costs during manufacturing stage can be lowered. ISM, with all the information from advanced sensor diagnostics, allows more flexible SOP. Successful implementation of QbD in the pharmaceutical industry requires a concerted effort between the regulators and the industry. The implementation of QbD is likely to result in increased efficiencies both for regulatory reviews and for pharmaceutical manufacturing. Real-time quality control will finally allow a real-time batch release.